Hello everyone, my name is Jessica Costa. I'm a PhD candidate in the Monogastric Nutrition Laboratory of Dr. Hans Stein at the University of Illinois, and today I will talk about the greenhouse gas emission, nitrogen balance, and ileal digestibility of amino acids and acid hydrolyzed ether extract in diets containing different inclusion rates of hybrid rye fed to growing pigs. So let's get started. Rye is a cereal grain valued for its agronomic advantages, including high drought and stress tolerance, winter hardiness, and the ability to thrive in low-quality soils. These characteristics make rye an ideal crop for farmers to grow in challenging environmental conditions. However, it's not commonly used as a feed ingredient for animals due to the risk of ergot contamination, fungal infection in the plant that can impair animal reproduction and growth. Recent advantages in the plant breeding have led to the development of hybrid rye varieties that are more resistant to disease, reducing ergot contamination. These hybrids also offer higher yields than conventional rye, making hybrid rye an available feed ingredient in animal diets. Here, I bring a comparison of the nutritional value between hybrid rye and corn, especially since corn is the most common cereal grain used in diets for animals in the US and in many parts of the world. Compared with corn, hybrid rye has less metabolizable energy. One reason for this is that it has less starch. Hybrid rye has about 60% starch, whereas corn has 63 to 65%. One thing that differentiates hybrid rye from other crops is its amount and composition of dietary fiber. Hybrid rye has approximately 19% total dietary fiber, of which around 20% is soluble fiber, which is more fermentable than the fiber in corn and other cereal grains. Corn, on the other hand, only has about 14% total dietary fiber, mostly insoluble and unfermentable. Likewise, hybrid rye contains less fat than corn, which also accounts for less metabolizable energy. In terms of digestible amino acids, hybrid rye has more total amino acids than corn, but the digestibility is reduced, resulting in similar content of digestible amino acids between hybrid rye and corn. However, regarding the first limiting amino acid in corn soybean meal diets, lysine, the standardized ileal digestible lysine of hybrid rye is 0.28%, which is higher than in corn, which contains only 0.18. Lastly, hybrid rye has more total phosphorus as well as greater digestible phosphorus. Therefore, as you can see here, the concentration of a standardized total tract digestible phosphorus is greater in hybrid rye than in corn. With all this information, it has been demonstrated that hybrid rye can be used as a feed ingredient in diets for pigs and can replace corn, which has been also demonstrated in previous research. Now, pork production is increasing, as well as there has been increasing concerns about the environmental footprint of livestock production. As we observe in this figure, the number of hog farms in the US described in the blue line has been reduced in the past decade, but the number of hogs per farm has increased, as shown in the red line, indicating that fewer farms are raising hogs, but those that remain are much larger in scale, suggesting a more intensive operation that can lead to greater environmental impacts. In fact, it has been reported that around 9% of the agricultural greenhouse gas emissions come from the livestock sector, which contributes to climate change. Therefore, strategies to reduce the environmental footprint of swine production are needed. In this regard, the emission of greenhouse gases by pigs can be influenced by several factors, including the composition of the diet. As an example, greater dietary fiber inclusion in diets for pigs has been associated with increased methane production due to greater hindgut fermentation, as well as greater nutrient excretion and reduced digestibility of nutrients such as amino acids and fat, which can result in lower feed efficiency of pigs and less growth performance. That said, although it has been demonstrated that hybrid rye can be used as a feed ingredient in diets for pigs, there is no information about how hybrid rye influences the production of greenhouse gases when fed to growing pigs, or if a change from using corn to using hybrid rye will result in a change in nitrogen retention or ileal digestibility of amino acids and acid hydrolyzed ether extract by growing pigs due to its different nutrient profile compared with corn. Therefore, two experiments were conducted to test the null hypothesis that greenhouse gas emissions, nitrogen balance, and the ileal digestibility of amino acids and acid hydrolyzed ether extract in diets are not affected by the inclusion of hybrid rye in diets fed to growing pigs. Moving on to the materials and methods, I will start with the diets. We used 3 experimental diets. A control diet was formulated using corn and soybean meal, and 2 additional diets were formulated by replacing 50% and 100% of corn with hybrid rye. All diets were supplemented with minerals, vitamins, and crystalline amino acids to meet the requirements for pigs. Experiment 1 took place at the swine calorimetry unit at the University of Illinois, where we used 24 pigs with an initial body weight of 52.38 kilograms that were housed in 6 metabolic chambers with 4 pigs per chamber. Each chamber was allotted to a replicated 3x3 Latin square design with 3 diets and 3 periods. Each period consisted of 13 days, where the initial 7 days were considered the adaptation period. For the next 6 days, feces and urine samples were collected from pans located in the bottom of the chambers, which allow us for total but separate collection of feces and urine using time-to-time procedure. These samples were analyzed for nitrogen, which was used to calculate the digestibility and retention of nitrogen. Likewise, each chamber has gas analyzers that allow us to measure oxygen consumption and carbon dioxide and methane productions for the same 6 days. The chamber was the experimental unit. For experiment 2, 12 ileal cannulated pigs with an initial body weight of 47 kilograms were housed in individual pens. Pigs were allotted to a quadruplicated 3x2 incomplete Latin square design with 3 diets and 2 periods. Each period consisted of 7 days, with the initial 5 days as the adaptation period and 2 days of collection. Ileal digesta was collected for 9 hours per day, as shown in the picture. In the experiment, the experimental unit was the pig. However, for both experiments, the statistical analysis was performed using the mixed procedure of SAS, with the diet as fixed effect and animal square and period as random effects. Contrast coefficients were also used to assess the linear effect of increasing hybrid rye in the diets. Now, moving on to the results, first I will set up my slides. Gray bars represent pigs fed the control diets, orange bar represents pigs that were fed the diet with 50% of corn replaced with hybrid rye, and blue bars represent the pigs fed the diet with 100% of corn replaced with hybrid rye. Results indicated that the average daily gain and the average daily feed intake were not different among pigs fed experimental diets, indicating that replacing 50 or 100% of corn with hybrid rye in diets for pigs did not impact growth performance. The same was observed in terms of the amount of oxygen consumed and the carbon dioxide and methane produced per pig, measured as liters per day per kilogram of body weight gain. This aligns with the observed growth performance data, indicating that under a production-based perspective, increasing levels of hybrid rye instead of corn in diets for pigs do not impact greenhouse gases. Even though the oxygen consumption and the carbon dioxide production were not affected by dietary treatment, we calculated the respiratory quotient by dividing the carbon dioxide produced by the oxygen consumed. The respiratory quotient was not different when pigs were fasting. However, it was reduced in the fed stage by increasing hybrid rye in the diet, because the carbon dioxide production was numerically decreased with the relatively constant oxygen consumption. This indicates that using hybrid rye in diets for pigs results in reduced carbon dioxide production relative to oxygen consumption. A lower respiratory quotient is associated with reduced carbohydrate utilization as an energy source in the body, indicating that pigs fed diets containing hybrid rye potentially shift their metabolic substrate to fat oxidation, excess amino acid catabolism, or using products of fiber fermentation. However, the lack of effects on the amount of greenhouse gas emissions indicates that nutrient utilization and metabolic efficiency were not changed by the substitution of corn with hybrid rye. Looking at the nitrogen balance data, we did not observe differences in the nitrogen intake absorbed and retained among pigs fed diets where 50 or 100% of corn was replaced with hybrid rye. Even though concentration of protein in hybrid rye is greater than in corn, the diets were balanced to meet the requirements for pigs, and therefore the excretion in feces and urine and the nitrogen utilization in the body were not affected by increasing hybrid rye in the diets. However, when we look at the apparent ileal digestibility, both dry matter and acid hydrolyzed ether extract were reduced when pigs were fed diets with increasing levels of hybrid rye instead of corn. Hybrid rye has more dietary fiber than corn, a soluble fiber, which can increase viscosity of digesta, increase endogenous losses, and reduce absorption of nutrients in the small intestine. Likewise, fiber may also interfere with bile acid formation and thereby reduce the digestibility of lipids. But hybrid rye contains less concentration of acid hydrolyzed ether extract compared with corn, which may also result in reduced digestibility. Likewise, it was observed that the standardized ileal digestibility of nitrogen and indispensable amino acids such as lysine and methionine were reduced when hybrid rye replaced 50 or 100% of corn in the diet, which can be attributed to the higher fiber content in hybrid rye, which may impact the digestion in the small intestine but can be fermented in the hindgut of pigs. In conclusion, replacing 50 or 100% of corn with hybrid rye in diets for growing pigs did not increase greenhouse gas emissions and did not impact nitrogen balance, although the ileal digestibility of amino acids and acid hydrolyzed ether extract were reduced. However, the growth performance of pigs is not impacted. Therefore, 100% of corn can be replaced with hybrid rye in diets for growing pigs. With that, I would like to thank the Monogastric Nutrition Laboratory and the National Pork Board for the financial support for this experiment. Thank you for listening to my presentation, and please visit our website at nutrition.nc.illinois.edu. Thank you.